Hi, all. Welcome back. We're going to finish talking about the history of anatomy and physiology. This is part two. I try to break things up to kind of 20-minute clips, um, just so you can sit down and watch them, and hopefully that's more convenient than a full-size lecture of 40 to 50 minutes. Um, but where we left off was the Renaissance shift. So at this point, when we're talking about the shift, what I'm talking about is this shift in relaxation of maybe religious expectations, in the sense that prior to the Renaissance, a lot of religious entities, to dissect a human, it was very taboo and was in many cases against the law. And after that, that kind of Renaissance shift really allowed for those dissections to occur. And on top of that, moving towards kind of more modern times, you also start to see this in embracing of the science. And so, a couple things. First, again, like I said, loosening of religious bans. Also, the rediscovery of some classical texts, so like doctrines that were made in China and India and Egypt, they were discovered and reinterpreted as kind of anatomy and physiology doctrines, and kind of that aspect of it. Okay? Human dissections, again, like I said, they return to medical studies. So for a good portion of time, hundreds, if not 1,000 years, it was taboo and it was not allowed in many regions to do autopsies, or to do even medical exploration of individuals who were terminally ill. And in some cases, it wasn't even legal to do a medical procedure, in some regions, to maybe save someone's life. This kind of, with the Renaissance shift, it kind of switched directions, and now a lot more places it was allowed to do, or it was not banned, at least. Okay, it might have still been taboo, and there might have still been quite a bit of pushback, but the regulations were lessened or loosened. Okay, and then one of the biggest, probably the biggest contributor to this huge output and really advances in anatomy and physiology during kind of the Renaissance time period. Um, so from about the 1500s, 1600s, that the big push was art. Right? And individuals started to realize the human body is a beautiful thing, and they started trying to combine the human body and science, and the human body, art and science. Sorry, and they started doing anatomical illustrations, actual illustrations, which either they were life-size illustrations or at least they were anatomically correct. They were in many different forms, from sketches to paintings to sculptures, but a lot of that, um, there's a big push to be anatomically correct. So no exaggerations of certain parts, and no misplacement of structures, and no exaggeration of size of certain parts. That everything was kind of to scale, even if it was just a sketch. And one of the big contributors to this, and probably one of the first kind of early pioneers of this, was Andreas Vesalius. And Andreas Vesalius, from 1514 to 1564, so not very many years, but remember, if you're not familiar with this, in the 1500s, the average lifespan was about 48, 50 years old. So about that for Vesalius. But he was really known as a very good anatomist. And part of that was his meticulous ability to dissecting out nerves and tendons and separating muscles and whatnot, really allowed him to write the book De Humane Corpus Fabrica. Um, and that was kind of like the base level, the background information, the background kind of book document with lots of sketches and things like that that paved the way for a lot of the other individuals that came after him to do their great work. And so again, one of the key components, because of that anatomical push, a lot of misconceptions from Galen, misconceptions from other early pioneers, they're erased. And it was Vesalis's work primarily early on that got rid of a lot of these errors, like misconnecting things and having things run in directions that they didn't actually run and all kinds of things. And so he used cadavers to kind of separate some of those errors. Some of the errors were honest errors from like Galen and a few of the others, because they were using animal models, not cadavers. And so they're basing everything on the animal and they're just extrapolating that to humans. And in some cases, you see that the two things are not the same. Okay? But along with greatness, as many individuals who might be at the top of their game, comes um, hatred. And Andres experienced this quite a bit. And not to say that he wasn't arrogant or have a big ego. He probably did have a big ego, and he probably was arrogant. Um, but he was one of the first, and he was good at what he did. He was a good anatomist. He had very steady hands, did very good dissections, and that really revolutionized anatomy. But in part, it took a while because there was quite a bit of hatred towards the individual because they felt that he wouldn't share his ideas, he didn't share procedures and methods at which he made these sketches and other things, illustrations, anatomically correct. Like he had distinct ways at which he would do it, and he wouldn't share those kind of things with anybody else. So William Harvey was building off of Andreas's work, and really he also has contributed with getting rid of or debunking a lot of the early misconceptions that were provided. Harvey discovered blood circulation, okay? And so Harvey went and using techniques, dissection, using kind of models that he developed, pumps and things like that. He developed a ability to discover that the blood is a circular system, it's a connected system. And so he wrote an entire book, De Motus Cordias, about the circulatory system. And so the overarching component of that book is that blood moves in closed loop and it's powered by the heart. And he got ridiculed for this big time. Because remember what we talked about last time, for about 1,000+ years, it was thought that the liver was what was powering the blood movement in the body. And now Harvey comes out and says, no, it's this heart structure, and here's how it works. And, you know, it was met with a lot of disagreement and not a lot of trust in Harvey's work. And then later, when he started showing, hey, this is how it works, and he could test things and started to use a little bit what we call the scientific method. He started designing questions and then experiments and then answering those questions based on those experiments. And that really started this revolutionary work in the circulatory system that Harvey kind of passed off, or at least his work was taken up by Marcello Malpighi. Okay? Malpighi, you're probably going to see both Harvey and Malpighi's names pop up again. Well, not probably, you are going to. When we get to the circulatory system, we start to talk about heart function and things like that. These individuals are really, really important for the circulatory system because they're discovering like how blood moves. And Malpighi was approaching it from a little bit different perspective than individuals in the past. Because of the invention of the microscope, Malpighi could start looking at the ends of veins and arteries, and he could be looking at capillary beds. And that's what he's most famous for, is observing capillaries and sketching the connection between veins and arteries and tracking them. And really, he took Harvey's work and completed the model of circulation. And so both individuals get a lot of credit for this, system that's very, very important, the system of circulation. And we'll talk a lot more about them. Then came the 1800s, and 1800s into the 1900s, and on and on and on, till today. Huge advances in science. And I would argue the 1800s, the value of the 1800s was the scientific method. Things that popped up in the 1800s that were really valuable to science, theory of evolution, which could tie these different components together. Okay? When individuals were dissecting things like frogs, and they're like, oh, look, a frog heart's different than a mammal heart. Why is there 3 chambers and they have 4 chambers? And then they dissect a fish and it's got 2 chambers. They started connecting the dots. These are what the chambers are used for. These are when, this is the difference between a bird's circulatory system and a mammal circulatory system. And what are the connections? What's the similarities? And all of that really was based on these 19th century advances. In addition, microscopes got way better. Okay? Technology got much better because you have the Industrial Revolution in the 1850s-ish era, and you have the invention of power. And now you can start making machines that can do things for you. And instead of everything being man-powered, now it can be powered by gasoline or some fuel type, coal, these different things, which really progressed the scientific advancements. And on top of it, we have the discovery of germ theory. We have the discovery of proteins. We have the discovery of DNA. All these things made a huge advancement in physiology for sure, but also anatomy. Okay? And so you get this rise of histology. Once you start making microscopes and you can section things and look at sections of all things, then your histological capabilities are much better. Biochemistry, okay, the ability to, run things through a machine and separate them based on their size, okay, like things like centrifuges and whatnot. All of this really advanced science. New instruments, microscopes, really expanded the knowledge. Okay, medicine. Okay, the discovery of penicillin and other things. Not just the ancient medicines that were going on from China and India and other parts of the world where they were using holistic approaches to, get at the physiology and the attachment to anatomy or between anatomy and physiology. Now you have these medicines that are being discovered and can be applied, and then you can look at experimental based physiology or physiological changes. And then, of course, modern. Um, I mean, it really just expanded from the mid 1800s till today. The medical advancements today are, I mean, they're amazing. Okay? Instead of the first procedure being cutting someone open, maybe the first procedure is, let's see if we can get an image of this to see if we even need to cut somebody open. These things are huge advancements in technology and science, and there's still much better advancements coming up, especially in the realm of molecular genetics. Molecular genetics is huge now, from the perspective of we we used to start, if there was some question about anatomy or physiology or both, we'd start at the organism level and then work our way down and get as close to the physiology as we possibly could. Uh, Prior to really the 1600s, it we didn't have really much for a microscope, so it was just whatever the naked eye could see. Microscopes were invented, and now you could start to see cells and things like that, and that connection. And then as we progress with molecular genetics, you're not looking at the cell, you're looking at the genetics, the DNA that's controlling for those proteins, that's controlling for the shape of objects. And really, that's allowing for a lot better look at form and function. And now, instead of, you'll hear me say it often, form or structure determines function. Well, now we can look at the genetics that determine the structure that determines the function. And so now you get three components that are very important. And so that really progressed us into being able to study cellular levels, and really being able to document flow from one region of the body to another, what systems are connected. Okay, that was huge, and still is. We're still trying to figure out all the potential proteins that a human body has, and all the potential proteins that your genes code for, and what are they used for. And I mean, every single day we're learning more about this. Okay? And of course, we have even a more kind of pressing issue now, is we have to think about the ethics. We have to think about, is it ethical to have a cadaver, to open that material up, to examine actual humans? How good are animal models? Is it even ethical to open up a chimpanzee or a monkey or any of these other organisms that we often used in the past as models for human um, anatomy and physiology? Is that ethical? And if it is ethical, how many? How many is necessary? And so a lot of those questions come into play. And we'll talk about those when we talk about some more examples of modern science. And also when we start reading some scientific literature, I'll point out, like, what's an IRB, what's an IACUC, and these different things, and who gets to decide whether your research is ethical or not. Okay. All right. Next time we're going to talk about anatomy and physiology, and really kind of the chemistry, just a little bit of chemistry, that is needed to kind of understand how cells work, how they communicate, those kind of things. And that's on the order for next time.